All right, I think we will conclude with our last speaker. It's my pleasure to introduce Sumayah Jamal, who's the CEO of ENB Therapeutics, a clinical stage biotech company that's taking a bold and highly differentiated approach to cancer immunotherapy. ENB is targeting a novel checkpoint, the endothelin B receptor, ETBR, which plays a key role in helping tumors evade the immune system. Their lead candidate, ENB-003, is a first-in-class selective ETBR inhibitor that has shown early promise in combination with PD-1 inhibitors like Keytruda, particularly in patients who typically don't respond to immunotherapy, such as those with platinum-resistant ovarian cancer or microsatellite stable disease. What's exciting is that ENB's work isn't just preclinical theory, it's backed by clinical data, support from Merck and Coherus, and collaboration with MD Anderson and the Cancer Research Institute. It's a compelling story built on strong science, meaningful clinical activity, and a smart and experienced team. Please join me in welcoming Sumayah Jamal.
Good morning, everyone, and thank you for being here today. My name is Sumayah Jamal. I'm CEO, co-founder, and chief scientific officer of ENB Therapeutics. ENB is a privately held clinical stage company developing treatments to supercharge immunotherapy. Our mission is to unlock new treatment options for drug-resistant cancer patients for whom there are few therapeutic options. Our therapeutic target is the ETBR, which has been implicated in anti-PD-1 failures. We're the first company to bring to clinical trial a selective ETBR inhibitor. In our completed phase 1 clinical trial conducted in collaboration with Merck, we demonstrated safety and efficacy in drug-resistant cancer patients. We are currently raising a Series B to support our phase 2 studies. Our primary phase 2 indication is platinum-refractory ovarian cancer and platinum-resistant ovarian cancer. This is a huge untapped market, and we anticipate an exit in 2027 once we achieve phase 2 clinical proof of concept. Our company underwent a very rigorous vetting process from both the MD Anderson Cancer Focus Fund as well as the Cancer Research Institute. This slide shows some of the feedback that we received from ovarian cancer KOLs. This very strong feedback provides validation for our company. This is our team. In terms of my background, I'm a physician scientist with a research career spanning over 30 years. I started studying the endothelin target in 1997 when I was a postdoctoral fellow. I am co-inventor on the first patents covering the endothelin B receptor as a therapeutic target for cancer. I started this company in 2015 to capitalize on the promise of the ETBR as an oncology application. And initially, I bootstrapped the company with personal funds, but have since taken the company through a venture-backed Series A round, entered into 2 pharma collaborations, and completed a successful phase 1 clinical trial. My co-founder and research mentor is Bob Schneider. He's a senior scientist at NYU School of Medicine, also a serial entrepreneur. He also led all of the tech transfer initiatives at NYU School of Medicine. My chief operating officer is Sandy Harm. She was at Merck for 24 years, where she served as U.S. Oncology Director of Commercial Operations and oversaw the development and launch of Keytruda. Our chief medical officer is also an industry veteran and very seasoned in leading clinical studies and FDA filings, and we're in the process of building out our advisory team. In terms of the problem that we're addressing, ovarian cancer is one of the many cancers that do not respond to Keytruda. ETBR expression predicts resistance to drugs like Keytruda, and the vast majority of ovarian cancer patients express ETBR, so it's no surprise that ovarian cancer patients do not respond to Keytruda. Expression of our target predicts very poor survival in ovarian cancer patient and also predicts cold tumors. So ovarian cancer patients that express ETBR, high levels of ETBR in their blood vessels, do not have tumor-infiltrating lymphocytes, which are required for drugs like Keytruda to respond. In terms of the patient population that we're targeting, it's the 40% of ovarian cancer patients that either do not respond to initial administration of platinum therapy or who relapse within 6 months of initial administration of platinum therapy. These patients are typically excluded from clinical trials because they're so difficult to treat. So this represents a significant unmet need. So our solution is our drug ENB-003, which unlocks the potential of anti-PD-1 therapy. And this slide shows you what ENB-003 can do when combined with Keytruda. So on the left are serial CT scans from a platinum refractory ovarian cancer patient. At baseline, this patient had a very large peritoneal tumor. This tumor was reduced by 68% at 6 weeks. It was undetectable by 12 weeks and remained undetectable out to a year. Overall, this patient had a 95% partial response that was durable out to a year. And again, this was a platinum refractory ovarian cancer patient. The patient additionally was microsatellite stable and PD-L1 negative. So this is a patient that is not expected to have a response to Keytruda. We believe that the addition of our drug, ENB-003, is what was responsible for this patient's remarkable response. In addition, the patient was initiated at the second lowest dose in our dose escalation study. We believe that if the patient had been dosed at a recommended phase 2 dose, that this would have been a complete response. On the right is a table comparing the combination of our drug ENB-003 plus Keytruda versus historical response rates to Keytruda alone. We were able to achieve a 40% objective response rate versus 8% with Keytruda alone, 80% disease control rate versus 37% with Keytruda alone, median progression-free survival of 12 months versus 2 months, and we also observed responses in patients like microsatellite stable patients and platinum refractory patients that have historical 0% response rate to Keytruda alone. ENB-003 is a first-in-class small molecule with very strong IP protection. In terms of its mechanism of action, the ETBR acts on step 5 of the anti-tumor immunity cycle. So this is upstream of where anti-PD-1 drugs work. So ETBR presents a block to anti-PD-1 activity. So our drug lifts that barrier and allows the downstream action of anti-PD-1 to take place. Ovarian cancer is a multi-billion-dollar market. However, this represents just the tip of the iceberg because our target, ETBR, is expressed in 40% of all solid tumors, which represents a huge untapped market. We have strategic collaborations in place with Merck and Coherus for our phase 2 studies. We're hoping to initiate those studies this year with data readout at the end of 2026 and a potential exit in 2027. Because of the high unmet need of our target patient population, we do believe that breakthrough therapy designation is possible after achieving phase 2 clinical proof of concept. We also have a potential companion diagnostic, which is a key differentiator between us and most immunotherapy drugs, for which there are no tests to select responder populations. We are raising a $17.5 million Series B financing. We have secured commitments for approximately 100% of our clinical costs, but need an additional $10 million to cover our operational costs. Use of proceeds for that $10 million is here. This provides cash runway through 2026, and it's a tranche raise which mitigates the raise. So to summarize, ENB-003 plus anti-PD-1 is a promising therapy for microsatellite stable cancers. We have observed very encouraging preliminary efficacy signals in platinum refractory and platinum resistant ovarian cancer in our phase 1 study. We have very favorable safety profile. No dose-limiting toxicities were observed in that phase 1 study. We have a potential candidate biomarker screen. We are hoping to initiate our phase 2 studies this year and hope for an exit in 2027. I'd like to thank the organizers for giving our company the opportunity to present today, and I'd like to thank you all for your attention.